Thank you for coming to my poster. I will be talking to you about Drosophila melanogaster as a model organism for nervous system development. So firstly, Drosophila melanogaster, aka fruit flies, are a great model organism, which just means that research findings can be applied to other organisms. So we took a look at the bone morphogenic pathway, which is evolutionarily conserved, and we looked at its role in the nervous system development during growth. So the nervous system is just the chemical and electrical messenger system of the body it uses to communicate. And the exchange of these messengers takes place at the synapse, which you can see here. And the synapse of interest that we looked at was the neuromuscular junction, or the NMJ. Now, this is because in flies, this synapse is a glutamatergic synapse, meaning glutamate is the neurotransmitter. And this is structurally and functionally very similar to the synapse in the central nervous system of humans, like the brain. So we look at the NMJ by doing an NMJ prep. So we'll take a third instar larva, which you can see here, and we'll fillet it open and stain it with fluorescent antibodies. Muscle will fluoresce green, nervous tissue will fluoresce pink. And then when you zoom in at an individual neuromuscular junction, you can see these button-like structures, which is akin to this in this diagram. And it just is a structure that houses the glutamatergic synapses. So in a normal wild-type fly, you'll see about 20 of these boutons in muscle floor. So the BMP pathway is partly responsible for how these structures grow and develop. And it is a retrograde signaling pathway, meaning the muscle sends messages to the neuron. So ultimately, the muscle sends a ligand or chemical messenger, and then it binds to receptors on the neuron. It becomes endocytosed into a vesicle. The vesicle complexes with other molecules and ultimately becomes a transcription regulatory complex, which affects DNA transcription. So we found that the protein kinase tau inhibits this BMP pathway, and a kinase is just something that phosphorylates things, which is a cellular on-off switch. So by removing an inhibitor, we'll see an overgrowth phenotype, which we can see with more boutons. So we'll have bouton counts over 20. Now, we have to see if it functions in the muscle or the neuron. So we used RNAi, and this essentially just degrades mRNA and it prevents the protein from being made. So that's how we accomplish loss of tau. And when we look at it, we see that it functions on the neuron side. This is because by removing tau in muscle tissue, it is the same phenotype as wild type, nothing changes. But if we remove tau in nervous tissue at the neuron, then we see the overgrowth phenotype. So it functions on the presynaptic side on the neuron. Now we know that tau has partners, which are also kinases in different tissues like the trachea, so GCK3 and tricornered. So we wanted to see if that was also the case at the NMJ. And we did the same exact method. We used RNAi, and we saw the same exact phenotype as loss of tau, which implicates that they are, yes, partners of tau at the NMJ. So once we knew that they were partners, we wanted to see the order in which they functioned, which is essentially called epistasis. So we used an overexpression background, which is overexpressing an inhibitor, which would cause an undergrowth phenotype, so bouton counts lower than 20. And then we paired it with loss of tau or loss of tricornered. So in the results, we can see that GCK3 overexpression, which was our background, that it's under 20, undergrowth phenotype. So that was our background. And then you can see in the pairs with tau GCK3 overexpression, the phenotype is almost wild type, which is slight undergrowth. So it's the same exact phenotype as GCK3 overexpression, meaning the overgrowth phenotype that we saw in tau, or loss of tau, gets suppressed, which means it functions upstream of GCK3. And then when we look at the tricornered GCK3 overexpression pair, that the undergrowth phenotype is suppressed and we see the loss of tricornered phenotype, meaning GCK3 overexpression is suppressed. And that just means that GCK3 functions upstream of tricornered. So the correct order in which they function is tau, GCK3, and tricornered. And that's largely what I did this summer. And thank you to everyone who helped make this possible. And thank you for coming to my poster talk.